All right. Today's topic is neurons and synapses. I've been struggling a little bit to get this video put together. So this is like, I don't know, take 12, hopefully. This is the winner. So anyhow, our essential understanding for the day is that neurons transmit messages, synapses modulate them. Neurons are nerve cells. They are sending these nerve impulses along their cell membranes, and then synapses are these connections between nerve cells, and they're modulating or regulating the message. So we're going to look first at the structure of a typical nerve cell, and then we're going to watch a YouTube video to get a nice overview of everything that's happening as we're transmitting these messages. So first up, structure of a nerve cell. These little ends here, these little branches are dendrites. Dendrites are collecting information from other nerve cells, from the environment, and then sending that information along the cell membrane of the nerve cell. This big chunk is the cell body, or the soma of the cell. Of course, it has a nucleus, and it's going to have endoplasmic reticulum and mitochondria, all those other good pieces. The cell body is going to kind of stretch out into this crazy long branch called an axon. So that nerve cell axon is just this long branch of nerve cell, which is surrounded by myelin sheath. Myelin sheath is made up of Schwann cells. And Schwann cells are full of lots of lipids, and they act like insulator just like on plastic insulation on copper wires. These Schwann cells are insulating the messages that are being passed along the axon. The axon is going to end in an appropriately termed axon terminal. Sometimes, especially if it's a motor neuron, and this one is a motor neuron, we might call the axon terminal the motor end plate. So motor end plate, axon terminal, same deal. Now that we have this generalized structure down, we're going to go ahead and watch this YouTube video, 3 minutes of your lives.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
Good. So let's look at these nerve impulses a little bit more in detail. So neurons or nerve cells are transmitting electrical impulses. We humans have about 85 billion neurons just in our brains, 46 miles of nerve cells all around our bodies. And nerve transmission is saltatory. So this word saltatory, if you're in Spanish, it's an easy one to remember because in Spanish, saltar means to jump. And saltatory is exactly that, this idea that nerve transmission is jumping along the axon. And so we're going to watch this one is much quicker, YouTube video about this idea of nerve transmission being saltatory.
You know what it feels like. You've been studying hard in med school, but it's your classmate who keeps getting high.
Saltatory conduction. Conduction through myelinated nerve fiber. Due to the myelination of neurons, action potentials may only occur at the nodes of Ranvier. Myelin is made up of insulating cells, which means depolarization cannot occur in myelinated regions. Between these cells, however, there are gaps known as nodes of Ranvier which are unmyelinated. As depolarization cannot occur at the cells making up the myelin sheath, the wave of depolarization can only occur at the nodes of Ranvier. Thus, action potentials appear to jump from node to node when traveling down an axon, and this phenomenon is known as saltatory conduction. And it serves as a means of increasing the rate of propagation of an action potential. Not only does saltatory conduction increase the speed of impulse transmission by causing the depolarization process to jump from one node to the next, it also conserves energy for the axon as depolarization only occurs at the nodes and not along the whole length of the nerve fiber as in unmyelinated fibers. This causes up to 100 times less movement of ions that would otherwise be necessary, therefore conserving the energy required to re-establish the sodium and potassium concentration differences across the membranes following a series of action potentials being propagated along the fiber.
Good. So again, nerve transmission is saltatory. The myelin sheath here along the axon is acting as an insulator, which means that all these details we're about to talk about, this nerve transmission piece, can only happen at these nodes of Ranvier. So the saltatory or saltatory conduction of the impulse, it's super fast because it's jumping. It's skipping over the myelin sheath, those Schwann cells. So what exactly is happening at these nodes of Ranvier? Well, first, we're getting to what we call the resting potential. A lot of physics, electricity stuff going on here in our nerve cells. So some interesting crossover between bio and physics. So our nerve cells are going to start at a resting potential, an electrical potential of about negative 70 millivolts. How that happens is this beautiful pump. And we know that if we have a protein pump, ATP is involved. This is active transport. So we're moving things against their concentration gradients. We have the active transport of first sodium. So we're going to send these 1, 2, 3 sodium ions outside of the cell. We're using ATP, lots of energy needed to move these things against their concentration gradients. This same pump is going to pull in 2 potassium ions. So I send 3 sodiums out, 2 potassiums in, 3 sodiums out, 2 potassiums in. And what happens is is a difference in the number of positive ions outside versus inside the cell. So if I have 3 positive things out for every 2 positive things in, the outside of the cell is more positive, which means that subsequently the inside compared to the outside is a little bit negative. We also have some chloride ions and some other things that are getting pushed in and out of the cell. The result is that the outside of the cell is more positive and the inside of the cell is more negative. And we can measure that negativity. It's about negative 70 millivolts. So this is the potential, electrical potential that we have due to these unequal concentrations of ions inside and outside of the nerve cell. So when a stimulus occurs, whether it's a pain receptor or a temperature receptor or neurotransmitters from another cell, we have this thing called depolarization occur. So in depolarization, we have these sodium channels. So this is considered a gated channel. So sometimes it can be closed and sometimes it can be open. When a stimulus arrives at the nerve cell, the sodium channel is going to open, and all the sodium that we pushed out of this cell can flow in. And so now that negative potential that we used to have is going away because I've got all these extra positive things flowing in. So all this sodium has flowed into the cell. I now have a positive potential on the inside of the cell. This is depolarization because we have unpolarized, we have flipped the charges inside and outside of the cell. But what's super cool about our nerve cells is that it doesn't start there. So right after depolarization, we have this process called repolarization. We're going to flip those charges right back. And so what happens is that sodium gate closes and this potassium gated ion channel is going to open. And all this beautiful potassium that we had been hoarding inside the cell can go out. And so the positive stuff that was inside is going away, which means that the inside of the cell flips from positive back to negative because of the loss of all that positively charged potassium. What's kind of amazing is that the pump, that sodium-potassium pump, is working to continue to push the sodium and the potassium back to the original sides. And so we don't have a complete depolarization of the whole cell and then a complete repolarization of the whole cell. But instead, little chunk by little chunk by little chunk by little chunk, those nodes of RON VA, between the myelin sheath cells are going to become depolarized and then repolarized and then go back to resting potential. But when this chunk depolarizes, it causes the next chunk to also depolarize. But while it's being depolarized, that first piece is going back to repolarization. This depolarization is going to cause the next chunk to depolarize while this part is repolarizing, while this first part has gone back to resting potential. So the nerve impulse definitely travels little bit by little bit all along the whole nerve cell. And we can actually measure that by poking our nerve cells with these tiny, tiny little baby electrodes. So we can stick electrodes into the cell. We can also measure the potential outside the cell, and then we can stimulate that nerve. And we can actually watch on the oscilloscope the potential changing along the cell. So we're starting at that negative 70 millivolts, that resting potential. The stimulus occurs and it causes depolarization. The cell gets more positive because all the sodium is going in. And then repolarization occurs. All the potassium is getting out and the cell goes back to a negative place. that graph you actually have to have memorized. So make sure that you can edit, annotate a graph of the action potential. Here we're starting at about -70 millivolts at resting potential. Here we have depolarization occurring as sodium ions are going into the cell. And then we have repolarization as potassium ions are getting out of the cell. And then we have this quirky little dip called hyperpolarization. Hyperpolarization, also known as the refractory period, before we get back to resting potential. And here, this little dip, what's happened is the sodium-potassium pump has started pushing that sodium that got in back out, but also all that potassium is out. And so we get a little extra negative until the pump, that sodium-potassium pump, is able to put the cell back to the normal resting potential of -70. You need to know all those pieces. So again, we have resting potential, depolarization as the sodium ions are getting in, sodium is getting into the cell, repolarization as the potassium ions are going out, and that refractory period as the pump is trying to put things right back into place. What's interesting about these nerve cells is that they are under constant stimulation. Things are always bumping us and stimulating our eyeballs. We have air molecules running into our cells, our skin cells all the time. And our brains are filtering out a lot of that stimulus. And part of the way that they do that is this idea of a threshold. So you might have a little stimulus, and the nerve cell just doesn't even care. But if you have a stimulus that gets us, that gets us up to this idea of threshold, that's when all the magic happens and the sodium comes in and the potassium goes out, and then we have to fix it. So that action potential is not going to happen every single time there's one little neurotransmitter or one little stimulus, but only if we can get up to threshold. And threshold is right at about -55 millivolts. So we're going to pause here and watch that video that we saw at the beginning again. If you feel like you know all these details, you can totally skip ahead 3 minutes. But for those of us who need the animation to kind of put all these details into place, here we go.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.
Good. If you have questions, definitely let me know when we video conference or send me an email. Read Cognity, watch the video again. But now we're going to skip. Oh, nope. Wait, I take that back. First, we are going to send our eyeball retinal nerve cells into a refractory period. So if you've played this game before, enjoy. If you've never done this to your eyeballs, I promise it is not dangerous. It's super fun. What we're going to do is stare at the plus sign in the middle of the flag for 30 seconds. You can totally blink, just don't look away from the plus sign for 30 seconds. We're down to 25 seconds already. Stare right at the middle of that plus sign. Don't look away from the plus sign. We're going to continue to look at the plus sign for 20 more seconds. For 15 more seconds, don't look away from the plus sign. Again, you can blink, but don't look away. Continue looking at the plus sign for 10, 9, 8, 7, 6, 5, 4, don't look away, 3, 2, don't look away. You should see the flag in real colors. Well, I mean like red, white, and blue colors. Yes, no, fun. Good. So what we did was overstimulate the yellow and green chemical pieces of our retinal cells, which allowed for the red and the blue to kind of overtake things when we're looking just at white because of that refractory period idea. So anyhow, you'll probably be seeing flags as we stare at these next couple of slides. But now we're going to flip over to the idea of synapses. So nerve cells transmit those messages, and then synapses are the guys that that are modulating or regulating those nerve impulses. So synapses are the junctions, these connections, these gaps between nerve cells, or between nerve cells and the receptor cells, maybe a muscle cell or something else. Here's showing an axon ending in that axon terminal. And then here is the dendrite of the next cell. We're going to call this the presynaptic, pre before the synapse cell, and this is the postsynaptic, post after the synapse cell. Here's, and then this is the micrograph. Of course, it's not quite as easy to see as when the drawing is over it, but that synapse, that gap is definitely in there. Here is that gap, that synapse. We can also see it in this picture if we zoom in on that axon terminal running into that dendrite. Here is the axon terminal. Here is the cell membrane of the dendrite. And then these are neurotransmitters that are being released from the axon terminal and are stimulating that next cell, the dendrite of the postsynaptic cell. So here's that picture one more time, just zoomed in super close. You can see that big, beautiful mitochondria. That cell needs lots of energy to make all those neurotransmitters. Neurotransmitters are filling all those vesicles here. And then the vesicles will release the neurotransmitters into the synapse, that gap between the two cells. The neurotransmitter chemicals will bind to the receptors on the membrane of the postsynaptic neuron, and things will happen. Here's another really pretty micrograph showing an axon terminal talking to a muscle cell. I know this is a muscle cell because of this structure down here. This is called a sarcomere muscle fiber. This is going to be the membrane of that postsynaptic muscle cell. And then this is the top, the very top of an axon terminal. So think of it not as the cell going this way, but instead the cell coming up at you from the screen. So this is the very top of that axon terminal, and we just kind of sliced it off the top. You can see some mitochondria here and here. And then again, all those beautiful vesicles that are full of neurotransmitters that are going to merge with the cell membrane, releasing all those neurotransmitters. The neurotransmitters will diffuse through the synapse here and then bind to receptors on the membrane of the postsynaptic cell. Here's another picture looking at the same thing. Here you can see the muscle fibers. Here we have the dendrites. If it were a nerve cell, but since it is since it's a muscle cell, they're not dendrites, it's just cell membrane. Here we have the cell membrane of the presynaptic nerve cell. Again, lots of vesicles full of neurotransmitters. Here we have mitochondria making all that energy. Here's the other side of the cell membrane of this nerve cell. And then this is some of that Schwann cell, that myelin sheath that's going to wrap all the way around this cell to provide that insulation. So what happens at this axon terminal? So here are all the steps that you need to memorize. So we have our action potential, that impulse that's been traveling along the axon. We finally get to the axon terminal, to the very end of the axon, and we've got sodium coming in, and we've got potassium going out, and sodium coming in, and potassium going out. And then we get down here, and instead of sodium and potassium, we're going to add yet another ion. Now I have a calcium channel. So that action potential, again, sodium in, potassium out, sodium in, potassium out, calcium comes on in. And that calcium is going to signal these vesicles that have been produced by the Golgi apparatus to take their neurotransmitters and bind to or dock with the membrane at the very end of the axon terminal. Exocytosis is going to occur. The neurotransmitters in the vesicle are going to be released from the presynaptic cell. We would call this the presynaptic cell because it's before the synapse. And then down here we have the receptors of the postsynaptic cell. And so those neurotransmitters are going to bind to the receptors, and then that impulse is going to cause whatever the neurotransmitter's job is to happen. So again, we have impulse coming along the axon. It's going to hit this new channel, the calcium channel. Calcium is going to come into the presynaptic cell right there at the axon terminal, is going to stimulate the vesicles that are full of neurotransmitters to dock with or bind to the cell membrane. Exocytosis occurs, allowing the neurotransmitters to be released. They diffuse across the synapse. The synapse, again, is this space between the two nerve cells. The neurotransmitters bind to the receptors, and things happen. So lots of different kinds of neurotransmitters. You've probably heard of a lot of these. So adrenaline and noradrenaline, these are produced by the adrenal glands that sit on top of the kidneys. Also called epinephrine and norepinephrine. This is going to increase heart rate, increase breathing rate, lots and lots of energy. This calms things back down. Dopamine is our reward our reward neurotransmitter. This is what gets us addicted to stuff. It's not good to mess around with your dopamine levels. Serotonin is also a feeling of well-being, but it's a calmer kind of content happiness. GABA calms us down. Acetylcholine helps us with learning. It also helps us with muscle contraction. Glutamate is another one involved in learning and memory, and then endorphins come along with exercise, being outside in the great outdoors, and just make us happy in general. And of course, you can imagine that if we are messing with these neurotransmitters, or if we have a natural imbalance of neurotransmitters, there are some disorders, psychological disorders that go along with those. And so acetylcholine can be involved. Imbalances in acetylcholine can be involved with Alzheimer's. Serotonin imbalances lead to all kinds of difficulties: depression, migraine, ADD, anxiety. Too much dopamine can lead to schizophrenia. Too little can be Parkinson's disease. GABA is related to epilepsy and glutamate, autism, some autism, not all autism, strokes and migraines. So lots of things related to neurotransmitters being out of whack. One of the things that we need to know about neurotransmitters and things that interfere with them is this idea of acetylcholine and acetylcholine esterase. So acetylcholine is one of our neurotransmitters. Again, it's involved with learning and memory. It's also involved with muscle contraction. So when a nerve cell releases acetylcholine, it is going to diffuse across the synapse, bind to the receptors on a muscle cell, and stimulate that muscle to contract. The muscle contracts. This enzyme called acetylcholine esterase. This is acetylcholine esterase. Acetylcholine esterase is going to break that acetylcholine down. So it's going to take it, it's going to chop it up into some pieces, and then those pieces are taken back up by the nerve cell and recycled to make more acetylcholine, so that later we can stimulate that muscle to contract again. And then the enzyme will break down the acetylcholine gets recycled, and then we can contract the muscle again. But if we are a bug and we're trying to eat a plant that has been treated with a neonicotinoid pesticide, then we are going to be blocking these acetylcholine receptors on the muscle cells. So for normal neurotransmission of acetylcholine, here again is our presynaptic neuron. Acetylcholine is released, it binds to the receptor. The muscle cell is going to contract. The enzyme breaks it down, it goes back into the cell, it's recycled, and then we can contract again, it gets recycled, we can contract again. But if we have neonicotinoid toxins in the way, these pesticide molecules look a little bit like acetylcholine. And so they're going to bind to the nerve cell receptors, the muscle cell receptors. But what's interesting about these neonicotinoid pesticides is that acetylcholine esterase, this enzyme, doesn't do a lick of good to break them down. And so these pesticides are going to bind to the receptors of the muscle cells and stay there forever, which means that it doesn't matter how much acetylcholine the bug releases, there's no more muscle contraction. The bug is paralyzed and it dies, which is great if you are a farmer trying to stop a swarm of locusts from destroying your crops. It's not so great if you are a human who needs the bees to pollinate the flowers so that you can breathe oxygen. So here you can see that as neonicotinoid imidacloprid use, this is an example of a neonicotinoid, is increasing, has increased over the last, 30 years. Bee colonies have been decreasing and they continue to decrease, and we're a little concerned about that. And that's all I got. So a couple fun things that you can do just to play with your nerve cells. So you can figure out how fast your nerve impulse travels. So we know that fastness or speed is distance divided by time. To find the distance, you're just going to use a measuring tape or something else, a ruler, whatever, to figure out the distance from your hand to your eyes. So whatever that distance is, I think mine was 114 centimeters, something like that. Anyhow, so that'll be the distance. And then for time, you're going to play this hand-eye coordination game. So how it works is when you're ready, you click that start button. The light's going to go from red to green. When it goes green, you click the button again like that. Whoa, that was really slow. And then you play it again and you get a nice average reaction time. And then you can take your distance from your hand to your eyeball, the length of that nerve cell, and then you can divide it by the reaction time, the speed at which things went. Now, since we're going from the eye to the hand, you just take that distance and divide, and that's your lab. If you want to read more about the speed of nerve impulses traveling in your brain, here's a really interesting article about how long it takes to think a thought. So lots of good science and then some guessing in here as well. So that's all I have for us today.